Hi. So, we will continue this antigen and immunogen property, and today I will talk about some more aspects of this antigen and immunogen, and then we will discuss about how we can elicitate antibody response in animal or human. Okay. So, let us begin this session. So, there is a term known as epitopes. Okay. So, what is epitope? Epitope is nothing. It's antigenic determinants recognized by B cells. Okay, and these antigenic determinants are also present for T cells also. So, all those antigenic determinants or surfaces over the antigen which is recognized by this B cells. B cells means the antibody expressing B cells, okay, and the T cells also. The processed antigen is presented along with MHC molecule, and this MHC molecule and this antigen is recognized by the T cell. So, these surfaces in case of antigen in case of B cells, we call it as the epitope, and the surfaces which is present over the antibody, okay, the surfaces which is interacting with the antigen, okay, the surfaces over the antibody which recognizes the surfaces on antigen, we call it as a paratope. So, just this is an important term. So, please remember that. Okay. So, B cell epitopes tend to be on the outside of antigen. So, you know that when I am talking about the antigen, I am particularly talking about the proteinaceous antigen. Okay. So, in three-dimensional structure, you may very well aware about that the surface of the protein, okay, the surface of protein is basically consists of hydrophilic amino acid, okay, and the inner core of the protein is basically consists of hydrophobic amino acid, okay, because there is no water molecule over there, okay, but on surface there are water molecule, and generally these are hydrophilic in nature, okay. So, the T cell epitope from protein is basically derived from enzymatic digestion. So, you remember that the antigen is taken up by the macrophages or dendritic cells, and then there will be action of enzyme in phagolysosome, okay, and then that digested protein turn to the peptide, and this peptide is associated with MHC class 2 if it is a TH cell. In case of TC cell, it's MHC class 1 molecule, okay. So, these molecules are presented along with respective MHC, and then that will activate respective T cells, okay. So, this is in case of T cell. In case of B cell, this is a naive protein or native protein, and this native protein is having some surfaces, and these surfaces are B cell epitope, okay, and the surfaces which is present on the antibody, we call it as a paratope, okay. So, there is a one example. So, glucagon is a basically hormone. When we immunize the animal with glucagon, then B cell and T cell epitope will be differ, okay. It is not the same epitopes, okay. For example, if animal is challenged with some antigen, the B cell epitope and T cell epitope will be entirely different, okay. So, B cell epitope have this characteristic property which I have already explained. Basically, they are located on the surface of antigen or immunogen, and basically it is accessible. So, since it is present on surface, so it is accessible by the antibodies, okay. For proteins, immunogens, the epitope can be, in case of B cell, the epitope can be a sequential or non-sequential epitope. Okay, I will explain you subsequent slide what I mean to say that, because you in previous session I have discussed that protein makes various level of a structure. Primary structure, which is a simple sequence. Okay, there will be a secondary structure. Secondary structure is they make some structure like a beta pleated sheet or alpha helices. Okay, and these secondary structure, several secondary structure which is made by particular or some sequences of the protein, this will make a tertiary structure, and this generally protein is present along with other protein in complex form, and this both these structures means the structure of one protein and another protein, that basically makes a quaternary structure. Okay, so let me explain what is the sequential and non-sequential epitope. I have a very nice scheme with that you can easily understand. So, sequential epitope recognized by B cell repertoire. So, here you can see that there is a one polypeptide chain, okay, which is represented by very colorful. You can see there is a yellow, and at both end, one end has a yellow color and another end has a blue color. So, you can understand this is a kind of sequence. Okay, and so the sequential epitope, so here you can see that the clone 1 can recognize this yellow and little orange color and darker orange color. So, this is a sequential epitope. Okay. So, if you see that various clone 1, 2, 3, 4, 5, 6, 7, 8, they are basically recognizing the sequence of this protein. Okay, and if you see the another structure, here you can see there is a non-sequential epitope. Okay, so when this polypeptide makes some structure, here you can see that this polypeptide basically making some kind of some structure, maybe there is a disulfide linkages, and that makes this loop kind of a structure. So, now the epitope is no more sequential epitope, it is a non-sequential epitope. Okay, here you can see that the clone 1 is basically recognizing the yellow and purple color. Okay, which is quite far. Okay, it is not in the sequence. Okay, and why this is recognizing yellow and purple, because there is some fold. Okay, so this kind of epitope we call it as a non-sequential epitope. Okay, so in general the protein which is inducing the immune response, particularly antibody response, the antibody is basically a mixture of it's a repertoire of B cell which is producing antibody, and these antibody have both kind of, they recognize both kind of epitopes, the sequential as well as non-sequential. Okay, so this is very important to understand, because there is one technique here, just I want to give you a note. Okay, so in general we think that this antibody recognizes the three-dimensional structure. Okay, so in this, if this is true, then when we perform the Western blotting, okay, if you have any idea about the Western blotting, so in Western blotting we separate the protein in denatured condition. Okay, we call it as we separate by using a technique known as SDS page. Over there we denature the protein and we separate it, and then we transfer it to the appropriate membrane, and then we probe with antibodies. Okay, so in that scenario the protein is completely denatured, so that doesn't have a three dimension means a native structure. So why antibody is reacting? So most likely, or in case of Western, so the epitope which is seen by this antibody is mainly the sequential epitope. Okay, so this is just a note that you should remember that the mixture of antibody which is generated against particular antigen, particularly proteinaceous antigen, that will this mixture of antibodies consist of both type which can recognize sequential as well as non-sequential epitope. Okay, so please remember this thing. This is very important. Now here I have a little comparison of antigen recognition by T cells and B cells. If you see, this will clear your further any doubt. Okay, if you have. So interaction with antigen in case of B cell, it involves it makes a basically binary complex okay, of membrane immunoglobulin, membrane bound immunoglobulin and antigen. So initially these B cells will have an antibody which is membrane bound, okay, and this membrane bound antibody is basically recognizing the native antigen, and this is a simple binary interaction. There is no involvement of other molecule, okay. But on another hand, the T cell, in case of T cell, it is basically a ternary complex, okay, ternary complex of T cell receptor. So T cell receptor is basically consists of two polypeptide chain. You will see in subsequent session that T cell in general, they have a two polypeptide chain which makes the T cell receptor, okay. So T cell receptor is basically consist of alpha and beta chain, okay. This TCR receptor is consist of alpha and beta chain, which is predominant form of T cells. There is some another kind of T cells which we call it as a gamma delta T cells. Those T cells have a gamma chain and delta chain, okay. Just forget about that thing. So generally T cell receptor is consist of two polypeptide chain, that is alpha and beta, and this alpha beta chain is basically interacting with MHC molecule, okay, and the peptide which is presented along with MHC molecule. So this makes a ternary complex. So here the interaction is quite complicated, I think you can understand, okay. Binding of soluble antigen, yes, in case of B cell they can bind with soluble antigen, but in case of T cell it is not at all true. T cell only see the processed antigen along with MHC class MHC molecule over the cells, okay. Either it will be antigen presenting cells or it will be a MHC class 1 expressing cells, any nucleated cells, okay. Involvement of MHC molecule in case of B cell, there is a no direct involvement of MHC molecule for interaction, okay, or for recognition, okay. But in case of T cell, it is most important. It requires the presentation of antigen along with MHC molecule. Without that, the T cell cannot activate or cannot recognize the antigen, okay. Chemical nature of antigen in case of B cell, mainly it is a proteinaceous, polysaccharide and very rarely lipids, okay. Lipids are, I have explained you that lipids cannot induce the good immune response, but when we want some immune response against lipid molecule, we tag it with the carrier molecule. So once you tag with this carrier molecule, then that can induce the antibody response, okay. In case of T cell, it is mostly the protein, but sometimes lipid and glycolipid is also presented on MHC like molecule. So I think I have told you in previous session, if you remember, there is a cell known as NKT cells, okay. These NKT cells basically recognizes the lipid antigen, okay, or lipid or glycolipids, okay. And this lipid and glycolipid is presented along with MHC like molecule known as CD1 family molecule, okay. And once they recognize, they are also inducing some immune response, okay. But in general, this is only the protein antigen, okay. Epitope properties. So, in case of B cell, this is accessible by BCR or B cell receptor, or BCR basically the antibody, okay, which is present over the B cell membrane, okay. This is accessible, okay, and as I told you, it's a mainly hydrophilic surfaces, okay, and mobile peptides containing sequential or non-sequential amino acid, okay. Now you can understand this sequential and non-sequential amino acid. In case of T cell, it is basically internal linear peptide produced after processing of antigen, and it is basically bound with MHC molecule, appropriate MHC molecule. It may be a CD1, it may be a MHC class 1, or it may be MHC class 2, okay. So, this is a comparison of antigen, okay, recognized by T cells and B cells. Now, let us move on. Now, I just want to, so you have understood about the antigen, you have understood about the epitope, okay, and now I want to show you what is the workflow for generation of monoclonal antibody or polyclonal antibody, okay. So, here there is a experimental animal. I have made it a mouse, you can use it as a rabbit. So, mouse is, I put mouse because I want to discuss about the monoclonal antibody, okay, which is mainly generated in the mouse, okay. So, here there is a mouse, and this is the antigen, and here you can understand that these 1, 2, 3, they are the surfaces, they are the antigenic determinant or epitope over the antigen. I made it this because it will help in understanding the concept of monoclonal and polyclonal antibody. So, when this mouse is challenged with this antigen, okay, then there will be after some time there will be a production of antibody which will recognize these surfaces, these antigenic determinant. Here for your convenience, I made a antibody molecules with same color, okay. So, one the green color is basically recognizing the epitope 1, the yellow color is recognizing basically epitope 2, and the dark purple color is basically recognizing the epitope 3. So, this is a polyclonal antibody, okay. Now, I have a challenge, I want to make an antibody which is only recognizing the surface 1, okay. So, if you take this polyclonal antibody and if you try to isolate this only one kind of antibody or which is recognizing epitope 1, it is technically very challenging to isolate that one kind of antibody, okay. So, in order to get one type of antibody, we adopt some another method. Here I am just showing the workflow, and after this I will talk about how to make the monoclonal antibody, okay. So, the workflow will be you immunize the mice, and when the mice is showing very good titer of antibody, you have to sacrifice this mice, okay. And after sacrificing, you take out the spleen, okay, and this spleen is basically containing the B cells, okay, which is making a particular kind of antibody, okay. Here you can see spleen has a 3 kind of these B cells, the green, the yellow and the purple, okay. So green will make this green color antibody, yellow will make the yellow color antibody, and so on. So, okay. So after making, you need to fuse these cells with myeloma cells. Okay. So this myeloma cells is basically a cancerous B cells. Okay. And when you fuse with this cell, then these cells means the green, yellow, and dark purple cells. Okay. So try to understand these cells, the green, yellow, and purple cell, they cannot multiply for longer time. Okay. Why? Because this is primary cell. After they may survive for few days, and after that they will die. Okay. So basically here what is the aim is to make the B cell line, okay, for particular cells. Okay. So in order to achieve this thing, we basically fuse these cells with cancerous cells. Okay. As here you can see that we will use the myeloma cells. Okay. And after that this fused cell can survive very long, or these cells become immortal. Okay. And once you have this immortal cells which can produce your antibody of your interest, okay, then you can expand those cells, okay, these particular cells, and then you can make a one kind of antibody. Okay. Try to understand, from polyclonal antibody, isolating one particular kind of antibody is extremely challenging. You can understand the molecular weight is same, only this is the affinity of the antibody with that particular epitope. It's a practically impossible, or I am not saying that it is practically impossible, but it is extremely difficult, okay, in order to isolate one kind of antibody from the mixture of antibody. Okay. But this is a very simple method. Okay. Here you basically fuse with myeloma cell, and then you grow these cells individually, then you can easily make this monoclonal antibody, one kind of antibody, and these antibody we call it as a monoclonal antibody. So now I will talk how to make this monoclonal antibody. In previous slide I just show the schematic or workflow. So basically what we do, we challenge the mice with your antigen of interest or protein of interest along with the adjuvant. I have explained you, we use the complete adjuvant or incomplete adjuvant. Okay. So after challenge, you basically check the titer of antibody. If the mice is showing very good titer of antibody, then you basically sacrifice and remove the spleen, and then you do further experiment. Okay. Here if the titer is very low, then you basically need to give the booster dose. Okay. You need to boost again with fluents, incomplete adjuvant, and antigen. And if the titer is good, then you can sacrifice and take out the spleen. Okay. So after that, what we do, we fuse with melanoma cell. Now here I will explain you the molecular mechanism for screening. Okay, how you will separate out all those cells which is fused with spleen cells, the spleen cell which is fused with melanoma cell. Okay. So this melanoma cell, this is basically derived from one knockout mice which we call it as a HGPRT. Okay. HGPRT is stand for hypoxanthine guanine phosphoribosyl transferase. Okay. So this mice is basically lacking this enzyme. I will explain you what is the use of this enzyme in subsequent slide. Okay. So this melanoma cell is derived from HGPRT knockout mice, and it is cancerous in nature. They can grow indefinitely. Okay. Just remember this thing. Okay. So now I will tell you how we do the screening. So the spleen cell which is coming from the immunized mice, okay, they have a property that they can grow for shorter time, okay, but melanoma cell can grow for indefinite time, right. So what we do, we basically we want all those cells which is fused with these spleen cell. Just for your information, we use various agents such as polyethylene glycol in order to fuse these two cells. Sometime people use Sendai virus, which basically fuses these cells, okay. And just for your I am just trying to refresh your biochemistry knowledge. So DNA in cells, the DNA synthesis is taking place by two major pathway. One pathway we call it as a de novo pathway, okay, and another is salvage pathway, okay. So in de novo pathway, all this basic units of DNA, that is the bases, the ribose sugar and inorganic phosphate is synthesized, and then this makes a nucleoside and nucleotide, and then this nucleoside and nucleotide polymerize, and that makes a DNA molecule, okay. This is a de novo pathway. In case of salvage pathway, you know that these cells are keep on dying, okay. In salvage pathway, this broken DNA or broken bases or nucleoside and nucleotide are used in order to make a new DNA molecule, okay. You remember that there is a semi-conservative method for DNA replication, right. Anyway, don't go in that detail. So let's so for this de novo pathway, there is a one very important enzyme is there which is needed in order to drive this de novo pathway. That enzyme name is dihydrofolate reductase, okay. So this dihydrofolate reductase is very important in terms of controlling this de novo pathway, okay. If you poison this pathway, then cell will die, okay, but it will not die immediately. This will switch to the salvage pathway. If salvage pathway fulfill their need for DNA replication, then it will not die, else it will die, okay. So there is a one molecule known as aminopterin. Aminopterin is basically a poison which inhibits this enzyme dihydrofolate reductase, okay. So this dihydrofolate reductase, if it is poisoned, then this will switch to the salvage pathway, and for salvage pathway you need this these bases, okay, like hypoxanthine, thymidine, in order to run this pathway. So what we do, we take this after fusion, we put these cells on a special media, okay, which we call it as a hat media, okay, which contains hypoxanthine, okay, aminopterin and thymidine, okay. So after putting this media, this hat media, what will happen then, this the cells which is basically so this aminopterin will kill the de novo pathway, okay. And please remember, the cells which is derived from spleen, this will have a both dihydrofolate reductase enzyme, as well as this will have a hypoxanthine guanine phosphoribosyl transferase, which is absent in melanoma cell. Please remember that thing, okay. So what will happen? So this, when you fuse and put it on hat media, then what will happen? This aminopterin will kill this de novo pathway, okay. Since this the hypoxanthine guanine phosphoribosyl transferase is also present in this cell. So only those cells will survive which is fused with a spleen cell. The fused cell means a spleen cell and melanoma cell which are fused, they will survive. Okay. So there will be a 3 or 4 situation. So one is that spleen cell will fuse with a spleen cell. Okay, this is first situation. When you will mix this spleen cell with melanoma cell, then there will be several situation. The first situation will be the spleen cell will fuse with a spleen cell. Okay, so those cells will die. Why? Because of because these cells will be inhibited in these cells. The DNA synthesis pathway is inhibited by aminopterin. This is first, and second reason is that these are primary cells, they cannot survive longer. Okay, this is a property of primary cells. Okay, another possibility is that melanoma cell will fuse with melanoma cells, so those cells will also not survive. Okay, because they will lack this HGPRT enzyme. Okay. The third situation will be the spleen cell will fuse with melanoma cell, and this spleen cell fuse with melanoma cell, they will survive. Why? Because they will regain the property of immortality because they are fusing with the cancer cell, number one, but they are lacking the since the melanoma cell is lacking HGPRT enzyme, which is provided by spleen cell, the spleen cell. Okay, then so this will have a HGPRT, the fused cell will have a HGPRT, and in media you have a hypoxanthine and thymidine. So this will trigger or activate this salvage pathway, and then they can survive. Okay, so in that way we screen those cells. So here you can see this is a very nice schematic for molecular basis for hybridoma selection. Okay, whatever I have explained you. Okay, so in that way we can isolate the hybridoma, the fused cell. Okay, spleen and melanoma cell, we also call it as a hybridoma. Okay, and then what we can do that we can also make a limited dilution. Basically what we will do, we will make dilution in such a way that each microvel titer plate will contain only one fused cell. Okay, so here you can see that one fused cells will be there. Okay, and this will be basically will amplify with time, and then we will test for the antibody. Okay, whether this is making our antibody of interest or not. Once you will find out, okay, this clone is making the antibody of interest, then you take that colony and expand it in bigger number, and then that will make a huge amount of antibody. So there are several ways by which you can make this antibody, expand these cells. One is that you take these cells and inject it into the peritoneum of mice. Okay, along with some agent. Okay, so then what will happen? This cancerous cell will grow like anything in the peritoneum, and that will make the belly of this mice extremely large, and you just aspirate that fluid, and this fluid will be rich in that monoclonal antibody. This is one way, but this is not very good way. Reason is that this will contain lot of other proteins. So if you need a very purified antibody, then this method is not good. The correct method will be you need to expand these cells in bigger flask and all those things, and then you can isolate the antibody. So that is better way. So this depends on your need, you can make your antibody. So now I have discussed about the polyclonal antibody and monoclonal antibody. Now I would like to tell the application of these two very important things. So here monoclonal antibody is basically used in basic research, diagnosis, therapeutic, and in basic research we basically use for this immunoblot, ELISA, okay, immunohistochemistry, flow cytometry. You do the cell analysis using this monoclonal antibody. It is widely used in diagnosis like pathogen detection, malignancy, diagnosis of array of infectious and autoimmune factors, okay. And in therapeutic, many monoclonal antibodies are used in therapeutic. We call it as a MAPS, okay, or bios. So they are quite emerging therapeutic agent, okay. They are also known as biosimilars. This can be used for autoimmune disease treatment, allergy treatment, and so on. So on another hand, polyclonal antibody is also used in basic research, for example, doing immunoprecipitation experiment, chromatin immunoprecipitation, immunoelectrophoresis, radial immunodiffusion, and so. It is also used in diagnosis, histopathology, and it is also used in therapeutic. So, one of the very good therapeutic application is generating the anti-snake bite serum, okay. So this is quite common, but in general for better results people try to use the monoclonal antibody, but there is some challenges with monoclonal antibody which is derived from mouse, because this mouse antibody is foreign to the human and that will cause the array of problem. So now there is a humanized antibody, and this has reduced the problem to larger extent, but still there is a problem. But I know that there are some companies, they made a mouse which can make a human antibody. They cloned the whole chunk of DNA in the mouse, okay. They replaced this antibody encoding gene segments, okay. So with this I will stop here, and in next session I will start more about the antibodies. Thank you.